Cerebrospinal fluid, or CSF, circulates freely through the ventricles of the brain, the central canal of the spinal cord, and the subarachnoid space. It is produced in each of the 4 ventricles by the choroid plexus, a structure that consists of specialized ependymal cells, their associated blood vessels, and connective tissues. The endothelial cells of blood vessels within the choroid plexus and ependymal cells are joined by tight junctions and collectively form the blood-CSF barrier, a portion of the blood-brain barrier. The selective permeability of this barrier controls what molecules are able to reach the cells of the brain. We can trace the flow of CSF starting in the lateral ventricles. From the lateral ventricles, CSF flows through the interventricular foramina to the 3rd ventricle. From here, the CSF enters the cerebral aqueduct and travels to the 4th ventricle. Once in the 4th ventricle, the fluid enters the subarachnoid space around the brain and spinal cord by either passing through the 2 lateral apertures in the walls of the 4th ventricle or through the single median aperture located in the roof of the 4th ventricle. A portion of the CSF also enters the central canal of the spinal cord. Accumulations of arachnoid villi, called arachnoid granulations, project into a venous channel within the dura mater known as the superior sagittal sinus. CSF is recycled into the venous part of the bloodstream at the arachnoid granulations. The turnover rate for CSF, from its production at the choroid plexus to its reabsorption into the superior sagittal sinus, is approximately 0.4 milliliters per minute.